Right. So in today's lecture, because you've been talking about white blood cells, you're going to be talking about red blood cells as well. A big part of red blood cells, of their function, of their structure, is hemoglobin. And you know about hemoglobin as a protein, because you've heard about that. But today I want to talk about the metabolism of iron, because iron is a major component of hemoglobin, and we're going to talk about the structure and metabolism of heme. Okay, so this is the topic of the lecture. It's going to be about heme and iron. We've already talked about ions, iron atoms, as prosthetic groups, as cofactors in various enzymes. Could you name any enzymes that contain iron in any form? Cytochrome C, absolutely. Do you know what form the iron is in? Well, it actually flips between iron 2 and iron 3, because that's how cytochrome C works as a transporter. But structurally, what form, how is the iron actually kept in the enzyme? Do you know, in cytochrome C? It's actually in heme, okay? Cytochrome C is a heme protein, contains heme. Any other enzymes that contain iron? Cytochrome C is technically not an enzyme, but yeah, it's a protein, yeah. Okay, this is a bit of a wake-up question, which I can see is not working all that well, but any other enzyme that contains iron? No? What about something in the respiratory chain? I mean, we've heard of cytochrome C, but cytochrome C technically is not an enzyme, but in the respiratory chain?
Iron and sulfur.
Absolutely. For example, in complex 1, we have iron-sulfur clusters. Okay, so that's another type, another form of iron that we can find in our body in different proteins. Okay, so that's not heme iron. It's not actually contained in heme, but it's there as well, and it actually serves a very important purpose. Okay, iron-sulfur clusters. And there are many other enzymes. Maybe I'll name another one from a completely different biochemical pathway. Have you heard of ribonucleotide reductase? Ribonucleotide reductase. What does it do?
Reduces.
Huh?
Reduces.
Probably ribonucleotides, you'd imagine. So what do we make if we reduce a ribonucleotide?
Ribonucleoside?
Nope. For that we need to add a phosphate. Well, we would have to actually take away the two phosphates.
Deoxyribonucleotide.
That's right. Okay, it's for the synthesis of deoxyribonucleotides. Very important for the synthesis of DNA. We can't actually synthesize DNA without ribonucleotide reductase. So anyway, that's another form of iron, another form of iron-containing protein where we don't have. It's not a heme protein. It doesn't have an iron-sulfur cluster. It actually has an iron oxide cluster or bridge. Okay, you don't have to remember that. It's just kind of an illustration of what different forms of iron can we have in our body. The reason, or at least part of the reason why we talk about iron in such detail is that iron is a very tricky element to work with. Does anyone know why it's tricky to work with? I mean, for organisms in biochemistry.
Because it induces oxidative stress.
Because it
induces oxidative stress.
Induces, yeah, very good. Okay, I thought you said reduces. Yeah, indeed. And which form of iron is especially dangerous? So which one? No, actually it's iron 2, okay? So Fe2 plus is very important for the induction of oxidative stress. And what is the name of the reaction in which it produces some nasty things? Does anyone remember? Fenton reaction, okay? Yeah, very good. So iron 2 is very dangerous because you can produce hydroxyl radicals from hydrogen peroxide, etc., etc. But there's something about iron 3 that also makes it a bit difficult to manipulate. It's extremely poorly soluble in water. Okay, at normal pH, at pH 7, it precipitates. It forms crystals. It forms just powder. It doesn't stay in solution. So in our body, we always have to keep iron either as iron 2, but that's pretty dangerous, or if it's iron 3, we have to bind it to something. It can't be just free in the body because it would immediately precipitate as iron hydroxide. Okay? So iron is difficult to work with because iron 2 is quite dangerous because it can cause oxidative stress, and iron 3 is very poorly soluble on its own, so we always have to bind it to something. Okay? And actually, even though iron is very abundant on Earth, there's actually a huge amount of iron everywhere, it's very difficult for organisms to get it from their surroundings so that they can use it. And the reason is that it's poorly soluble. So all the organisms, including bacteria, actually have to devise quite complicated mechanisms how to extract iron, how to make it more available, and then use it for the metabolic purposes. So bacteria actually synthesize these very complicated compounds that are called chelators, iron chelators. You heard chelators before? Chelator? Chelator. So it's a substance that forms a chelate. And a chelate is a complex. Okay, it's a special type of complex. Basically, a chelate is a complex where the ligand has more than one binding site. That's a chelate. Okay, does it make sense? Really?
So
Sure? The ligands, yeah. So if the ligand has at least 2 binding sites, so it binds at least with 2 bonds onto the central atom, then it's called a chelate. Okay? So anyway, these chelators are synthesized by bacteria, but we also have our own chelators in a way, although they are proteins. And why am I talking about it? Because in pharmacology, some of these bacterial chelators are used to treat people who have too much iron in their body due to some diseases or some other conditions. And we're going to talk about iron in the second half of the lecture in more detail. Right. So as I said, most of the time, the iron in the body is bound to something. And one of the major forms in which it's bound is as heme. Okay? Now, heme is a fairly large polycyclic compound that when you look at for the first time, it looks very complicated. But actually, the structure can be broken down into simple building blocks. And we're actually going to go through the building itself, so you're going to see what the building blocks are, which makes the structure, at least in principle, fairly simple. So the molecule of heme is actually composed of 4 rings. Those are 5-membered rings containing nitrogen. What is the name of the ring? That's the name of the whole ring, which I still haven't actually drawn, but what is this? Huh? Yeah, okay. Pyrrole. Okay, so those are pyrrole rings. Can you all see that? And because there are 4 pyrrole rings in heme, we also call it heme belongs to a set of tetrapyrroles. Okay, so heme is a tetrapyrrole. Just means that it has 4 pyrrole rings. Now, there are several different types of tetrapyrroles, and they can be either linear or they can be cyclical. Okay, and we're going to see some of the linear ones later on. Okay? So tetrapyrrole is a term that I want you to remember. Okay? It's anything that contains, it's a substance that contains 4 pyrrole rings, but it either can be cyclical or it is linear. So in the case of heme, it is a cyclical tetrapyrrole. So we actually have methylene bridges. So these are CH2 groups that connect the ring all together. And heme is also an unsaturated ring. So I'm going to put in all the double bonds in the correct places. You won't be asked to draw the structure, okay, just so that you don't freak out. Okay. So when I draw it like this, what can you see about the structure? Is there anything interesting?
Is it conjugated?
Very good. It is a conjugated system. All the double bonds are conjugated. Okay? What does it tell you when there are lots of conjugated double bonds there? It's a large ring with lots of conjugated double bonds. What does it tell you about the properties of the compound? Anything interesting? Yeah?
They're more stable.
Yeah, absolutely. It's going to be more stable than if the bonds were not conjugated. Absolutely, that's true. And there is another property which you can see immediately if you look at the compound. It's going to be colored. Okay? And the reason why I mention it is that most people think that the reason heme is red is because of the iron, that it's the iron that it contains that makes it red. But actually, most of the color comes from these conjugated double bonds in the whole heme molecule. Okay? Obviously, the iron atom actually just modifies the wavelengths which it absorbs. Okay? But the color actually comes from these double bonds. Okay? Right. This ring, as was already mentioned before, is called porphyrin. Porphyrin. Okay? So porphyrin is a cyclical tetrapyrrole. Yeah? So this is porphyrin. Now, there are two things that we have to add. There are two things that we have to add to porphyrin to make it into heme. One of them is iron. Okay? So we put iron, let's say it's 2+, which is going to be bound to the 4 nitrogens. Does anyone know if there are porphyrins in nature that contain a different metal in the middle than iron?
Bilirubin.
Huh?
Bilirubin.
No, bilirubin is actually not a porphyrin. It's a tetrapyrrole, but it's not a porphyrin. It's not cyclical, it's linear. And we're going to talk about it in a minute. Anybody? You heard of chlorophyll? Chlorophyll? What is chlorophyll? It is in plants. And what is it for? What does it look like?
It is for photosynthesis and production of oxygen.
Okay, yeah, so it's important for photosynthesis, it's important for absorbing light, for all the processes. And the structure of chlorophyll actually looks very similar. It is a porphyrin, but it contains magnesium in the middle. Okay, so it doesn't have iron, it's magnesium there. And therefore it's green, because the magnesium actually shifts the absorption. Okay, so while heme is red, magnesium is green. Okay? But again, the color actually comes from these electrons. The metal ion just modifies which wavelengths it's going to absorb. So anyway, one of the things that we need to add to porphyrin is, as you correctly said, is iron. And the other thing is, or are, substituents on these rings. So we can put different substituents on these rings to form different forms, different kinds of heme. Okay, and I'm going to show you in the synthesis how we actually make these different kinds of heme, at least one of them. Okay, so these substituents are various groups that are attached to these rings, and this, with the specific substituents, this is heme. This is the structure of heme. Okay, as I said, I'm going to show you what these substituents are, but at this point, this is just the general structure of heme. Okay, does it make sense that it's actually not that complicated? Yeah. Okay. Any questions? No. Let's now have a look at how heme. Yes.
Sorry, just roughly. So it's the ion that is in the middle part and the groups attached to the porphyrin that
Yep.
will result in heme?
Yes. Yeah. Okay, let's look at the synthesis of heme, which is, considering that the structure looks fairly complicated, the synthesis is actually relatively simple. it's simple chemically, but there are a lot of new names, okay? So the new names of compounds might become quite confusing, but the synthesis is actually relatively simple. Compared, for example, to the synthesis of purines, it's much simpler than that. Okay. So the whole of heme molecule is built from only 3 building blocks. Okay? One of the building blocks is iron, obviously, but this comes at the very last step. So iron is put into heme at the very, very last reaction. Okay? So we're going to leave that aside. The other 2 building blocks that we actually use to build the rest of the molecule are glycine and succinyl coenzyme A, succinyl CoA. Well, actually. Okay, glycine and succinyl CoA. In the first step of the reaction, we basically join the 2. You can imagine that we remove this carbon dioxide away. And we just stick the 2 molecules together as they are written. Okay, so we'll obviously also release coenzyme A. We stick the 2 together. So what we get is something like this. Okay, do you see how we've just joined the 2 together? Yeah? Yeah, I know, it's really exciting, isn't it? Okay, so this is the first step in the synthesis of heme. This compound is called 5-alpha-labulinic acid. 5-aminolabulinic acid. 5-aminolabulinic acid. Or sometimes you can find delta-aminolabulinic acid. It's usually abbreviated as ALA. Okay? So that's the first step. Where do you think this first step is going to occur? Just looking at the substrate.
Mitochondria. Mitochondria.
Why in mitochondria?
Succinyl coenzyme A is part of the fblt.
Okay? So succinyl CoA is an intermediate in the Krebs cycle, so this is where we have it. The first step of heme synthesis occurs in the mitochondria. Okay? This is fairly important because then all the other steps, except for the last 3, occur in the cytosol. So the beginning and the end of heme synthesis is in mitochondria, but the middle is in cytosol. Okay? I'm going to go through all the steps just so you realize that the first step and the few last steps are in mitochondria. The rest of it is in the cytosol. Okay?
Is it also in the mitochondrial membrane? Or?
Sorry?
Is it also in the mitochondrial
No, it's actually in the matrix. Okay? The first step is in the matrix. Okay? The enzyme that catalyzes this reaction is called ALA synthase. So 5-aminolabulinic acid synthase. And ALA synthase contains vitamin B6, pyridoxal phosphate, as its cofactor. Okay, pyridoxal phosphate. You may recall, I think we talked about pyridoxal phosphate, that often it's used when you do something with amino acids, when you decarboxylate amino acids, when you dehydrate or whatever you do with amino acids, usually you have pyridoxal phosphate as a cofactor. Okay, so here it binds glycine and joins it to transaxanyl-CoA. Okay, so that's the first reaction of heme synthesis. Now, in the second reaction, the second step, we take 2 molecules of ALA and we join them together. Okay, and I'll show you how. Okay, so I'm just going to redraw basically 2 molecules of ALA next to each other so you can see how they're joined together. Okay, 2 molecules of ALA next to each other. Yeah, can you see that? Okay, so in the next step we take 2 molecules, we take away 2 molecules of water, and once again we join the 2 molecules of ALA together, and we join them in such a way. Okay, so this oxygen, these two hydrogens go away as one molecule of water. And this oxygen and these 2 hydrogens go away as another, in a very simplified way. Okay, and what we get from it, it's going to maybe it's going to become clearer when I actually draw the results. Something like this. Yeah, can you see how the 5-membered ring is formed? Yeah.
Can you explain once again which molecules that goes away as water?
Well, in the 2 molecules of delta ALA, you take this oxygen and these hydrogens, and this oxygen and these hydrogens. Again, that's a bit of a simplification. The actual chemistry is a bit more complicated, but let's imagine that it works that way. Okay? Because you can see that the double bonds are actually shifted around a little bit different way. Okay, the resulting compound, as you can already see, that is the actual building block of heme, which is going to basically put 4 of them around it. Okay, this is the pyrrole, and here is the methylene bridge between the pyrroles. So we just basically join them together. That's all we're going to do, pretty much. Okay, with some other few steps. This resulting compound is called porphobilinogen. And usually it's abbreviated as PBG. Porphobilinogen. You don't necessarily have to draw all the structures, okay? It's better if you just pay attention, because you can find all of this in any biochemistry textbook, anywhere on the web, okay? The synthesis of heme is just everywhere, so you don't have to actually draw every atom, okay? It's very easy to find the reaction scheme, okay? Right, what would be the name of the enzyme? What do you think? If you were to guess, what is the name of this enzyme? No. Okay, so it could be a porphobilinogen synthase that it synthesizes, yeah. But if we start with the substrates? Dehydratase, okay, because we are dehydrating, not dehydrogenating. We are dehydrating, we're removing water. Yeah, makes sense? So the name of the enzyme is ALA dehydratase. It's removing water from ALA. Ala dehydratase. Okay, so now we basically have the building block from which we can easily see how we can build the whole molecule of heme. And for that, we just need to pay a little bit more attention to these two substituents. Okay, I told you that to the porphyrin ring that we saw, we need to add iron, but we also need to add some substituents to the rings. Okay, and here we see the beginnings of these substituents. How would you call these groups? Anybody? Okay, strictly in organic chemistry, you'd call them carboxymethyl and carboxyethyl. Okay, those are the groups. But actually, for historical reasons, they're called kind of in an opposite way. So this is called acetate, and this is called propionate, because it looks like acetic acid, and this looks like propionic acid. Does it make sense? Okay? So we usually mark them as A and P. And you're going to see that something is going to be happening with those substituents. But it's important to realize how A and P are basically arranged on this one building block. Okay, it's going to be clear in a minute why it's important to realize that. Okay, so any questions so far to the synthesis? No? No? Okay, so I'm going to erase that so that I can show you the next step. All right, so now as I said, it kind of makes sense to take 4 molecules of porphobilinogen and just join them next to each other. Okay? So what we get is a linear tetrapyrrole, which looks something like this, in a very schematic way. Something like this. Ish, okay. Don't draw that, yeah?
Only the joining of these two is it happening now in cytosol?
Yes,
yeah.
yes. So we've moved ALA, once we've made ALA, we move it out of the mitochondrion, and the rest of the steps are, now we're already in the cytosol. Actually, with ALA dehydratase, we're already in the cytosol, okay? So just the first step, and then as I said, some of the last steps as well are in mitochondria. Right, so we have something like this, okay? What are the substituents look like? Well, they're going to be always A, P, A, P, A, P, A, P. Okay, as you would expect. Okay, this is just a schematic, okay? It doesn't actually look like that, but you can easily find the actual structure. Right, this molecule, as I said, is a linear tetrapyrrole, but it's not a porphyrin, okay? So porphyrin has to be a ring, it has to be unsaturated. Okay, so this is a linear tetrapyrrole, and it's called, again, with a not very helpful name, hydroxymethylbilane. Hydroxymethylbilane. Okay, so with 4 molecules of porphobilinogen, we join them together, we make hydroxymethylbilane. Okay? The enzyme is called porphobilinogen deaminase because we need to remove this amino group in order to join those molecules together. Okay? So the enzyme is porphobilinogen deaminase. Sometimes you can find also that it's called hydroxymethylbilane synthase, but the more usual name is porphobilinogen deaminase. Okay? Makes sense so far? Okay, what do you think is going to be the next step?
Forming the ring?
Exactly, we join them, we cyclize we basically close the ring, okay? Well, one way of closing the ring would be to close it basically in a very simple way. Basically, as we see the molecule, like this. So in the ring, we would have, again, AP, 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 AP. Okay? We just take this and join it together. Now, this molecule is called uroporphyrinogen 1. I'll explain why that complicated name. Uroporphyrinogen 1. Uroporphyrinogen 1. Okay? So this is what you get if you just take hydroxymethylbilane and make a ring. Okay? And indeed, for this reaction, you don't actually need any enzyme. It happens spontaneously. But, and that's a big but, we can't actually make heme out of this. This is not the right starting point for heme. We need to make a slightly different uroporphyrinogen, which is called uroporphyrinogen 3, which looks almost the same, but not quite. Because in uroporphyrinogen 3, the substituents are AP, 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 PA. So one of the rings is flipped around. Can you see the difference? This ring is basically, in the uroporphyrinogen 3, is flipped over. You see that? Yeah?
Sorry?
Raise your hand if you can see that. Okay, even those who weren't looking can see it. Excellent.
But these two reactions, they weren't, how do you say, they weren't related, were they?
Well, they're sort of related, but
Yeah.
this is a spontaneous reaction. This is a reaction that requires an enzyme. And this is the reaction that we want. This happens spontaneously, but we actually want this reaction, and there is an enzyme for that. Okay? And the enzyme is called uroporphyrinogen 1 synthase. Yeah, it's really confusing. The enzyme that we want is called uroporphyrinogen 1 synthase, but in order to make uroporphyrinogen 3, we also need a uroporphyrinogen 3 co-synthase. Okay? So in order to make uroporphyrinogen 3, we have an enzyme that's called uroporphyrinogen 1 synthase, which just basically joins the ring. But in the presence of uroporphyrinogen 3 co-synthase, we make uroporphyrinogen 3. Okay? Raise your hand if you got that. Okay, I was quite surprised. I'm not sure I got it. Yeah?
So in order to make uroporphyrinogen 1, we don't need the enzyme, but we still need the enzyme to close the ring for uroporphyrinogen.
That's right. So uroporphyrinogen 1 can either form spontaneously without any enzyme, or it can form if you just put in uroporphyrinogen 1 synthase. Because the uroporphyrinogen 1 synthase will just close the ring. That's it.
Why do we need it if it will close anyway without this?
Yeah, but without the enzyme, it will always close as uroporphyrinogen 1, which we don't need. But in the presence of the enzyme and the co-synthase, we make this uroporphyrinogen 3, and that's the one that we want. Does it make sense?
And we'll...
Really? Yeah? Good.
And will we also get some uroporphyrinogen 1 just...
Probably, yeah, a little bit. But it will get broken down or just excreted in urine or whatever. But not a lot, because obviously a lot of these syntheses are really well regulated so that all the intermediates go where they need to go. So under normal conditions, you don't get a lot of uroporphyrinogen 1. Co-synthase. So it's uroporphyrinogen 3 co-synthase. Okay, it's just an addition to the synthase so that it makes uroporphyrinogen 3. For those who are interested in organic chemistry, it's quite interesting to find in a textbook, but you have to find a really good textbook, where they actually show the chemistry of how this works, because flipping the ring is actually a very it looks simple, but it's actually super complicated. You have to do some really strange things to it so that you can flip it. It's a very interesting reaction. Anyway, yeah.
Because one again show us where is the difference between.
Sure. So in the usual, both in hydroxymethylbilane and in the not very useful uroporphyrinogen 1, you just go symmetrically, basically. You just go AP, 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 AP around the ring. Okay. Here in uroporphyrinogen 3, you go AP, 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 but here it's PA. So this ring is flipped around. Okay. It's not symmetrical anymore. Yeah.
But this flipping happens after the cycling?
It happens basically before at the same time. Let's say at the same time. Okay. It happens at the same time. It's part of the reaction. But as I say, the actual steps are very interesting. It's a very complicated reaction. Okay, so this is one of like the main bits, or one of the important bits in the synthesis. The rest is really simple. Okay, so if you understand that we have to make uroporphyrinogen 3, which looks different, it's a different isomer. After that, it's fairly simple. Okay, so everybody's clear about this strange reaction? Yeah, good. So once we have uroporphyrinogen 3, we need to make two things. We need to, well, three things actually, to the molecule in order to make heme. We said in the beginning that the iron is the very last thing. Okay, so again, let's leave adding the iron to the last step. The other two things that we need to do is add some more double bonds. Okay, there already are some double bonds, I'm just not drawing them, but we need to add more in order to make a conjugate system. So here, uroporphyrinogen 3 is still doesn't have a conjugated system of double bonds. Okay, there are some single bonds present, so they break down the conjugation. So uroporphyrinogen 3 is not colored. Okay, it's a colorless compound. Okay, hydroxymethylbilane is colorless. Porphobilinogen colorless. Okay, they're not colored. Now, how can you tell that the ring is not fully saturated? In the name, when you see porphyrinogen, that means that it's still not a porphyrin. Okay, so any porphyrinogen doesn't have a conjugated system. And once we make porphyrin from porphyrinogen, that's when we actually oxidize it into a proper conjugated system. Make sense? Yeah? So whenever you see porphyrinogen, it's not going to be colored. Okay? Or could be a little bit, could be a yellowish or something, but it's not going to have a strong color, and it doesn't have a fully developed conjugated system. Okay? Right. So one thing that we have to do is to close the ring. Not close the ring, but actually close the conjugated system. But the other thing is we actually have to do something with these substituents. Because in heme, at least in one type of heme, which is heme B, and that's the one we're going to be talking about, these substituents are actually decarboxylated. Okay, they're decarboxylated in the synthesis, and they are decarboxylated in such a way that what do we get if we decarboxylate this acetate group? Okay, we get a methyl. Okay, so all the acetates are decarboxylated to methyl. Right? What do we get by decarboxylation of this group? We don't actually get ethyl. That would be too easy. What we get is get a vinyl. Looking like this. Vinyl. It's a double bond. Okay? A vinyl group. Okay, so in the synthesis of heme B, we decarboxylate all the acetyl groups to methyl. And then we decarboxylate 2 of the propionate groups to vinyl. Just 2 of them, of the 4. Okay? So in the final molecule of, well, still not heme, but almost heme, all the formally acetate substituents are methyls, and 2 of the propionyls are vinyls.
Are we now after the making of more double bonds?
No, that's actually before. Okay, so it's still called a porphyrinogen. It's actually called coproporphyrinogen, but anyway, you don't have to know that. Okay, so first we decarboxylate the substituents, and then we add the double bonds. Okay, and that already happens back in mitochondria. Okay.
Both of them?
Actually, the last decarboxylation and the oxidation happen in mitochondria. But you don't have to be all that specific, okay? If you remember that the oxidation of the ring and then the adding of iron is in mitochondria, that's going to be enough, okay? Yes?
Did you say the decarboxylation occurs only on 2A and 2B?
No.
All four, or
All the A's
only?
and 2 of the P's, okay? And after the decarboxylation of the A's, the intermediate is called coproporphyrinogen. Coproporphyrinogen. You don't have to remember that intermediate, okay? But it's called coproporphyrinogen. And once we decarboxylate the propionates, it's called protoporphyrinogen. Okay? So now we have protoporphyrinogen, and we oxidize it in mitochondria. We oxidize protoporphyrinogen to protoporphyrin, and that's an actual porphyrin. Protoporphyrin, yep. Protoporphyrin.
And that's after decarboxylation, then?
After decarboxylation, it's called protoporphyrogen, and then we oxidize it and we make protoporphyrin. Protoporphyrin. And protoporphyrin is basically heme without iron. Okay, so in the very last step of the synthesis, as I said, it's in mitochondria, we add iron to it, and we make heme. Okay? So protoporphyrin is heme without iron. Okay, and that's the whole synthesis of heme.
Can you please repeat that this happened now again in mitochondria?
Yes.
Okay.
Let's suppose the adding of more double bonds and the decarbonation.
The last decarboxylation, yes. So from coproporphyrin to protoporphyrinogen, that's already mitochondria.
Still, the adding of double bonds happened after the decarboxylation.
That's the last but one step, yes. So it's here. Here it's oxidation, okay? So here we are in, well, coprogen to protoporphyrinogen. Here we are in mitochondria. Okay? Again, every textbook has it. Wikipedia has it. Everybody, okay? So it's very easy to find the synthesis. Okay, any questions? No? So before we take a very, very short break, let's talk a little bit about the degradation of heme, how we actually break it down. Okay? Because the heme needs to be broken down, especially from red blood cells, because red blood cells, as you probably know, don't live forever. They actually live relatively short time. So once they are broken down, all the heme from hemoglobin has to be broken down, and we have to do something with it. The degradation of heme is much simpler than the synthesis. What we do is, we take heme and we oxidize it by an enzyme called heme oxygenase. To a product, to a linear tetrapyrrole. So we basically break the ring open. Okay? And the new tetrapyrrole, which is a linear tetrapyrrole, is called biliverdine. Biliverdine. Okay? Biliverdine. The other two products of this reaction, what would you think are the other products? So we crack the ring open.
Remove the iron.
Very good. So another byproduct is going to be iron. It's then reused, and we're going to be talking about that a little bit later on. But the third product is very interesting, because heme oxygenase actually breaks the ring by oxidizing one of the methylene bridges. Okay? So it takes, basically, it rips one of the bridges out, which opens the ring. Okay? Now, normally in biochemistry, what would you expect to happen when you oxidize carbon? You get usually, carbon dioxide. Okay. Here, however, you make carbon monoxide. Okay? So in our body, constantly cells that are breaking down heme are making carbon monoxide. So even if you don't live in a city and you don't smoke, you're always going to have a certain level of carbon monoxide in your blood because it's being produced by heme oxygenase. Okay? Quite an interesting reaction. And actually it's been hypothesized that carbon monoxide can serve as a signaling molecule, the same way as NO. But I think it's still quite controversial. Okay, biliverdine is then reduced to bilirubin, and bilirubin is then transported in the liver, and in the liver it's conjugated with glucuronic acid. Okay, you know glucuronic acid from xenobiotic metabolism and detoxification. No? Glucuronic acid? Raise your hand if you've heard of glucuronic acid. Excellent. Okay. So, when I ask that you've heard of it, just nod or something. Okay. So bilirubin is then conjugated in the liver, and then it's excreted where? In the bile. Okay. So it goes into the GIT, into the intestines, and it's excreted out from the body. Okay. So that's basically the whole degradation of heme. All right? Any questions?
The iron who is removed, you said we need to bind to something?
Yeah, and we're going to talk about it in the second half, okay? What actually goes on with the heme. Very last thing, clinically, how do we call a condition where we have too much bilirubin in the body, in the blood? Okay, jaundice or icterus. Yeah, very good. There are many kinds of jaundices which we usually classify based on where the problem is. And we classify them as prehepatic, hepatic, and post-hepatic. Okay, where the problem is. So prehepatic jaundice is usually caused by when too much heme is broken down, especially red blood cells. Okay, so when many red blood cells are broken down, too many, so that the liver can't actually cope with excreting all the bilirubin, you can get a prehepatic jaundice. Make sense? Yeah, there's just too much bilirubin, liver can't really cope with it. It's prehepatic jaundice. The other possibility is hepatic jaundice, so the problem is at the level of liver. Liver is damaged, so it can't actually excrete enough bilirubin, and therefore it accumulates. Very often it's caused by infections, by viral infections, by viral hepatitis. Okay? And finally, post-hepatic jaundice, what would the cause be there? What do you think could cause it? That's right. If the bile ducts are blocked, okay, often by bile concrement or by a tumor, then we obviously get an accumulation of bilirubin in the blood. Okay, so 3 kinds of jaundices. I'm not going to go into detail, but it's just important to mention that. All right, 5-minute break, and we'll try to finish it off after that. Okay, I know it was a fairly short 5 minutes, but we still have quite a bit to go through. Do you have any questions about either the synthesis or degradation of heme? Is there anything that's not entirely clear? No? Okay, as I said, it's very easy to find in any textbook or on the internet. So if you have any questions, you can probably find the answers there. All right, let's now talk about the metabolism of iron itself. Because we know that we need iron for the synthesis of heme, we need it for many other processes. We also get it from the degradation of heme. So we need to see how iron is trafficked in the body, how it's absorbed in the body, how it's regulated. Speaking of regulation, I should probably mention something about the regulation of heme synthesis as well. It's going to be fairly quick because it's relatively simple. The synthesis of heme is regulated in most cells by feedback inhibition. Okay? The rate-limiting step, the most important regulatory step in the synthesis of heme, is the first step. So it's ALA synthase, is the enzyme that is regulated. And as I said, in most cells, there's a feedback inhibition by heme on ALA synthase. So if there's plenty of heme, ALA synthase is blocked, and therefore the cell doesn't produce any more heme, which makes perfect sense, presumably. Okay? However, there's one tissue where this could be a problem. Which tissue could have a problem if there was a feedback inhibition by heme? Because most cells don't want to produce any more heme that they, require for their enzymes. There's one tissue which needs to produce a lot more heme than...
Is it muscle? No.
Not really?
Red blood cells?
Red blood cells, okay? Red blood cells need to pack themselves with hemoglobin that requires huge amounts of heme, okay? So there, the feedback inhibition wouldn't really work because they couldn't really, make a lot of hemoglobin. So therefore, we know that there are two isoenzymes. There are two forms of ALAS. ALAS1 is the one that we find in normal cells, and that one is feedback inhibited by heme. Then there's ALAS2, which is expressed only in erythroid precursors, in the precursors of red blood cells in the bone marrow. And this one is not regulated by heme, but it's actually regulated by the availability of iron. Okay? So if there's not enough iron, ALAS2 is inhibited, which again makes sense because we don't want our red blood cells to produce huge amounts of protoporphyrin when we cannot actually put iron in there, okay? Because that would be dangerous, okay? So that's about regulation. The other thing I did mention is that there exist quite a few defects of heme synthesis. And the diseases, the group of diseases are called porphyrias. Porphyrias. They're called porphyrias because it's a very ancient name, because porphyros, apparently in Greek, means purple or dark red. And the urine of these patients is usually dark red or purple. The reason is that the various defects in the enzymes of heme synthesis can lead to accumulation of some of the intermediates, and those intermediates can spontaneously form porphyrins that are, as we agreed, are colored. Okay, so these patients with porphyrias, I'm not going to go into details. There are many different types of porphyrias. Actually, produce these strange, aberrant, incorrect porphyrins that are then excreted in the urine, and the urine is colored, and that's what the name of the disease. You will learn quite a lot about porphyrias later on, especially in internal medicine. It's a big group of diseases, and for that, if for nothing else, it's quite useful to know the steps in the synthesis of heme, because every individual enzyme can have a defect, and it produces a different kind of porphyria. Okay, so if not for your enlightenment, then for actually learning internal medicine, it's very useful to know the synthesis of heme, okay, and the steps that are there. Last thing about defects in heme synthesis, ALA dehydratase, the second enzyme, is a metalloenzyme. It contains zinc, and there's zinc in its active site. It requires zinc. However, when people are poisoned with lead, yeah? Okay? Lead poisoning. People are poisoned with lead. Lead very often swaps with zinc in many different enzymes. And if it swaps with zinc in ALA dehydratase, it blocks the activity of the enzyme. And why is it important? Well, because in these people who are poisoned with lead, we can find accumulation of ALA, because ALA dehydratase is blocked, so there's accumulation of ALA. And we can actually measure the concentration of ALA in the urine as a measure of lead poisoning. Okay, so it's a marker of lead poisoning to measure ALA in the urine because lead blocks ALA dehydratase. Okay, so that's just another clinical.
How can we remove the lead?
It's a good question. I mean, there are special chelators for lead removal, so you can try removing it that way, but usually the best way is just to stop the people from ingesting more lead, because usually those are chronic intoxications, so they get it from, you know, very old plumbing, which is now very, very rare, or if they have lead paint somewhere, okay? So usually what you do is you try to remove the people from the environment where they get more lead in, okay? Because it's rarely acute intoxication, it's usually chronic. But yes, there are drugs that basically can attempt to remove lead from the body. Okay. Right, okay, so that's all about heme. Let's go and talk about iron. There are two parts of the iron chapter. One is how iron is absorbed and regulated in the gut, in the gastrointestinal tract, so how we actually absorb iron from the diet. And the other part is how our cells in the body regulate and absorb iron from the blood. Okay? Two different ways of getting iron in. So if this is an enterocyte, okay, most of iron is absorbed in the duodenum. Okay, so this is a duodenal enterocyte. Okay, so that's the nucleus. And iron is absorbed from the gut by a specific transporter, which is called DMT1. DMT1 stands for divalent metal transporter. Okay, DMT1, divalent metal transporter. And as the name suggests, we need the iron to be in a divalent form. So it has to be as iron 2+ in order to be absorbed. Again, it sort of makes sense because we know that iron 3 is very poorly soluble. Okay? Now, how do we reduce iron 3 that is in our diet? How do we reduce it to iron 2? Well, there are two things that help. One is the low pH in the stomach. Okay? Because in low pH, it's much easier, the redox potential changes, it's much easier to reduce iron from iron 3 to iron 2. So the low pH in the stomach helps reduce iron to iron 2. Okay? So people who have, for example, who take antacids, who take drugs that basically increase the pH in the stomach, that block the production of acid, will have decreased iron absorption. Okay? Yeah. So anyway, so we have DMT1 that transports iron 2 ions into the cell. Divalent metal transporter. Divalent metal transporter. Right, so we transport iron, ferrous iron into the cell, and there are several things that can happen. One is that if there's too much iron, it gets stored in a protein called ferritin. Oops. Ferritin. Now, ferritin, usually in textbooks, you find that it's a storage protein. So you imagine that basically the iron goes in the protein, and whenever the cell needs it, it can go out of the protein. Actually, ferritin is more of a detoxifying protein or protective protein. Because actually, once the iron gets into ferritin, it's very, very difficult to get it out again. Actually, the whole ferritin has to be completely destroyed and digested in a lysosome in order to get the iron out. So I think it's better to think about ferritin as where you put all the iron that you don't need anymore. Okay? So it's usually considered a storage protein, but yeah, it's very, very difficult to get it out, get it back out. Ferritin is, for those who are really interested, it's a very interesting molecule. It's actually a spherical, a pretty large sphere that has an envelope made of protein, okay, by protein subunits. But in the center of it, you get a crystal of iron hydroxide. Okay, so it's a tiny little sphere of crystallic ferric hydroxide that is surrounded by protein. That just basically explains even more that getting the iron out is very difficult because you get a mineral. You get a. Have you heard of magnetite as a mineral? Magnetite? No, it's a magnetic form of iron oxide. Anyway, so you have basically little bits of magnetite in your body that are just surrounded by protein. That's ferritin. Anyway.
I will go back to 3+ when it
Yes, it's oxidized. It's oxidized in the ferritin. Yeah. Yes.
So is everything stored in the enterocytes then?
That's one possibility. Okay. But it actually only happens when there's just too much, and I'll explain the regulation in a minute. So that's one possibility. The other possibility is that it's used in the enterocyte. For example, it goes into mitochondria and it's put into heme. That's another possibility. But the important possibility, and that's the one we're really interested in, is that it's exported from the enterocyte into the blood. Okay? That's because we're absorbing it from the diet. We obviously wanted to push it into blood. There is another transporter on the abluminal side of the enterocyte, which is called ferroportin. Ferroportin. That's the transporter, ferroportin. Yeah. So we have 2+ now on the abluminal side. Okay? But as we said, we don't really want, or our body doesn't really want to have just free iron floating around. It needs to bind to something. Okay? And the transporter for iron that is used in our body is called transferrin. Transferrin, usually abbreviated TF. Transferrin. Now, transferrin binds 2 atoms of iron, but it binds 2 atoms of iron 3. So the iron needs to be oxidized first in order to bind to transferrin. So we have iron 3 here. And the oxidase that we use for it, a lot of new names, is called hephaestin. Hephaestin.
This is the enzyme?
That's the oxidase that oxidizes, yeah. I mean, those of you who know Greek mythology, you probably know where the name comes from. No? Hephaestus, the blacksmith of the gods. Yeah? Okay. Yeah. So here you can see how inventive scientists can be. Anyway, so hephaestin. Yes?
So is that found in blood or in the enterocyte?
It's on the enterocyte. Okay, so it's on the membrane and it oxidizes iron 2 to iron 3 so they can bind to transferrin.
And transferrin is found in the blood?
In the blood, yeah, that's right. It's in the plasma, yeah. Okay? So transferrin is the transporter of iron in the plasma, in the blood. Yep. Okay, good. So let's have a look what happens when a cell needs some iron. Okay, so this is in the gastrointestinal tract. This is the absorption from the diet. Now, what happens if a cell in the body needs iron? So this is our generic cell. Well, anyway. And if any cell of our body needs iron, it expresses transferrin receptors. So transferrin is a fairly large protein, which carries always 2 atoms of iron 3. And if a cell needs iron, it expresses transferrin receptors. Okay? What do they do? Well, obviously, a transferrin receptor will bind transferrin, but it will only bind transferrin that contains iron. Okay? It will not bind transferrin without iron, because obviously in the blood we'll have both. We'll have both transferrin with iron, so-called saturated transferrin, or we can have transferrin without iron, which is sometimes called, well, often called apotransferrin. Apotransferrin. Okay, that's the name for transferrin without iron, empty transferrin, apotransferrin. So anyway, transferrin receptor, do you want me to slow down a little bit? Yeah. Yeah? Okay. Okay. So there are 2 forms of transferrin. saturated transferrin, fully saturated transferrin, and apotransferrin, so one without iron. There's actually a form of transferrin that only contains one iron, but let's not complicate things, okay? The transferrin receptor has a very high affinity for saturated transferrin, so it will not bind apotransferrin. Okay, it will become clear why I'm saying that, okay? So, the cell needs iron, it expresses transferrin receptors bind transferrin, and what happens is that the whole complex of transferrin receptor and transferrin is endocytosed. So we have an endocytic vesicle that contains the transferrin receptor with transferrin, okay, bound to it. Now, transferrin has an extremely high affinity for iron. It binds iron extremely strongly, okay? So at normal conditions, you can't actually get the iron out. It's a very, very strong chelator of iron, okay? However, that only is true at normal pH, at neutral pH. So what happens in the endocytic vesicle, we have proton pumps that pump protons into the vesicle that decrease the pH. Okay? And as the pH is decreased, the iron from transferrin is released into the vesicle. Okay? Because the affinity of transferrin is much lower at low pH than it is at neutral pH. Okay? So iron 3 is released.
Can you please repeat everything from the endocytosis?
Yeah, so it's endocytosed. Then the vesicle contains, well, it actually fuses with other vesicles, but it doesn't matter, with proton pumps. And those proton pumps pump protons into the vesicle, and thereby they decrease the pH, they acidify the vesicle.
And
Okay.
why do they have to do that?
Well, because the affinity of transferrin for iron is extremely high at neutral pH, but it goes down very steeply at lower pH. Okay? So because we need to release the iron from the transferrin, and the way to do it is by decreasing the pH. So we decrease the pH in the vesicle, we release iron 3. The iron 3 is reduced in the vesicle to iron 2, and it's not entirely clear how. Okay? So it's reduced to iron 2+, and then it's exported from the vesicle into the cell by our friend DNT1. Okay, so we lowered pH, released iron 3 from transferrin, we reduced it to iron 2, not entirely clear how, and then it's transported out of the vesicle into the cell. And here in the cell, once again, it can be used for example, the synthesis of heme in mitochondria. It can be put into ferritin. All sorts of things can happen to it. Okay? Now, what happens with transferrin and with transferrin receptor? Well, they're both recycled back to the membrane. But since I mentioned that transferrin receptor has basically no affinity to apotransferrin, to transferrin without any iron, the transferrin is then released back in the blood. Okay? So this whole complex is then recycled back to the membrane. And since without the iron, which we released here, the affinity of the receptor to transferrin without iron is very low, the transferrin is released from the receptor and can go away and can be refilled anywhere where there's iron. Yep.
So when the H plus gets pumped in,
Yep.
it's only the iron that releases, not the ferritin.
Transferrin.
Transferrin, I mean.
Yeah, no, the transferrin is actually still bound to the complex. And that's again, that at low pH, and I don't want to confuse you, but at low pH, the affinity of the receptor to apotransferrin is high. So at a low pH, they still stick together. But once they are exposed to normal pH, they release from the complex. Okay? I didn't want to confuse you now, but yeah, but anyway, that's how it works. Okay, so this is the so-called transferrin cycle. Okay? The cells express transferrin receptors, they're taken up, acidified, iron's released, and then the whole thing is recycled again. Okay? Now, how are these two processes regulated? One regulation needs to be at the level of absorbing iron from the diet. Okay? As we said, iron can be very dangerous. Okay? So our body is always trying to very tightly regulate how much iron we're getting in. Okay? Iron is essential, so we have to get in some iron, but it's also dangerous, so we can't actually get any more. So it's very tightly regulated how much we get. Do you know what is the recommended daily allowance of iron? About 10 milligrams for males and about 20 milligrams for females in fertile age. Because obviously menstrual bleeding is one of the most important losses of iron, physiological losses of iron that we can get. Because otherwise, the only way we lose iron is basically by shedding cells. Okay? So from your skin cells, from your hair, from your intestinal cells, that's the only way how we can lose iron. Okay? Otherwise, there's no way to excrete it normally. Okay? But as I said, menstrual bleeding obviously is a major loss of iron. Therefore, women who are menstruating need to get more iron. Okay? So it's about 10 for males and about 20 for fertile females. Right. But as I said, the amount that is absorbed, because the amount of iron in our modern diet is actually very high. There's a lot of meat, there's a lot of dairy products, so the amount of iron is very high. Therefore, we need to regulate how much we get in. And the regulation of this is actually carried out on the level of ferroportin. Okay? The amount of ferroportin in the enterocytes is regulated by a hormone called hepcidin. Hepcidin. Hepcidin is a hormone that's released from the liver in response to high levels of iron. If there is enough iron, hepcidin is released from the liver and it decreases the amount of ferroportin in enterocytes. So it removes ferroportin from enterocyte. Okay? What happens when that happens? Well, we still get absorption of iron into the enterocyte, but it cannot be pushed into the blood because there's no ferroportin there. And since you know that enterocytes usually live for only about 3 days, the enterocyte that has plenty of iron inside, as ferritin or whatever, is just released into the stool, and we don't care about it. Okay? So the regulation is on the level of ferroportin via the enzyme called hepcidin. Okay? Good. What about the regulation at the level of the cell? Yep.
Was hepcidin an enzyme or a hormone?
It's a hormone. Yep. Did I say enzyme? Yeah? Okay, it's a hormone. Yeah. Okay, what about at the level of an individual cell? How does the cell regulate how much iron it gets? Well, first of all, there needs to be a sensor for iron. We need to have some protein that senses how much iron is available. Okay? And that's actually necessary for the secretion of hepcidin as well. The liver has to have a way of knowing how much iron there is. Okay? Let's have a look how it works on the level of a generic cell, not necessarily a liver cell. Are you still following me? Okay? Look a bit. Okay, good. The sensor for iron in the cell is called IRP, which stands for iron response element binding protein. Just stop me wherever it's too fast or it doesn't make any sense. IRP binds to a sequence called iron response element. However, normally when you see a protein that binds to a nucleic acid sequence, it's a DNA sequence. And it's influencing basically the transcription of DNA to RNA. Okay, that's the usual regulation of protein amounts. Okay, you have a transcription factor that influences the rate of transcription. Okay, Here, however, the IRE is a sequence of RNA, of mRNA, and the regulation is at the level of translation. So it's not a transcriptional control, it's translational control. Okay? So IRP is a sensor of iron, and it influences the translation of various proteins, not the transcription. Okay? Now, how does it work? Well, first of all, how does IRP work? How does it sense how much iron there is around? Well, IRP is actually an analog, or homolog, or whatever you want to call it, of the enzyme aconitase. Which pathway is aconitase important for?
TCA.
Yeah, for the TCA cycle, for the Krebs cycle. Okay, aconitase is one of the enzymes. Now, aconitase contains an iron sulfur cluster. Okay, there's an iron sulfur cluster. And the iron sulfur cluster, one of the iron atoms in the cluster is fairly labile. It can leave the cluster and bind back. Okay? So whether the iron atom is in the cluster or isn't depends on the concentration of iron around the enzyme. Does that make sense? Raise your hand if that makes sense. Okay? So you have a iron sulfur cluster, like so, iron here, okay? Sulfur, 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 okay? Now one of these atoms can leave or come back from the cluster, okay? Now, if there is a lot of iron around the enzyme, most likely the iron will be in the cluster, okay? Because basically the likelihood is higher that it will bind to the cluster. If there's no iron around, the likelihood is that the iron will leave, just based on basically the movements of ions. Okay? Does it make sense? Yeah? Raise your hand now if it makes sense. Good. And this is exactly how IRP senses iron levels. It has an iron sulfur cluster similar to aconitase. It basically is aconitase, okay, at least one of these subtypes. When there's too little iron, the iron leaves the cluster, and as the cluster disintegrates, it changes the conformation of the protein. Okay? And once that happens, IRP can bind to IRE on mRNA. Okay? So this is your mRNA. There's a 5' end and 3' end. I'll explain what these weird things are. Okay? Here is our gene that's to be translated. Okay? And these weird mushrooms are secondary structures that are formed by IRE. So those are IREs. Iron response elements. Okay? Yep.
Are they nucleotide sequences?
Yeah, this is mRNA. This whole thing is mRNA.
Yeah.
And this bit of the mRNA is a sequence called IRE. And it forms this loop on the mRNA.
Okay, and the IRP binds to?
That's right. Okay, I'm going to show you how that works.
Can you shortly repeat the iron is released when no iron is around?
Yes.
In the plasma?
Yeah, not in the plasma. We're talking about inside the cell.
Okay, inside the cell.
Okay, it's inside the cell. So when there's too little iron inside the cell, the iron from the iron sulfur cluster in IRP is released, which changes the conformation of the protein, and now the protein can bind to IRE. Okay?
IRP are just floating around inside the cell? Or?
Pretty much, yeah. Okay? Actually, so that I don't confuse you, I'm going to do something like this. Okay. So this is a gene for, this is an mRNA for some protein, okay? It could be transferrin receptor or some of that protein. It is a protein. Now, is it getting too fast or too...
Time-wise.
Oh, okay, okay, we're almost done, okay. If IRP is activated, it can bind to IREs, okay? So imagine that in this specific gene, we bind IRP to these IREs. What happens to the translation of this mRNA?
Increases translation?
Why would it increase translation?
Maybe where's the promoter? Or...
Well, they will be in transcription. Okay, but this actually blocks translation because basically the ribosome can't really bind onto this mRNA because there are these proteins that block it. Okay, so if the IRE is present or are present at the 5' end, the binding of IRP will block translation and will therefore decrease the amount of the protein. Okay, this is true, for example. Well, let's try to figure it out. Which protein would we need to decrease if there is not enough iron? Very good. We didn't actually get there. But yeah, allosynthes would be definitely one of them. And indeed, allosynthes has IREs. Okay, remember allosynthes? We don't want allosynthes in erythroid cells to be active if we don't have enough iron. Okay, so by this mechanism, we can suppress the translation of allosynthes in those erythroid cells. Okay, so very good. The other possibility would be ferritin. We don't need to make ferritin if we don't have enough iron. There's no point making new ferritin. Okay, so that's one possibility. Okay, and I'm going to be super quick. The other possibility is that the IREs are at the 3' end. Okay, what happens there if we bind IRP? Well, since you remember that mRNA degradation starts from the 3' end. Yeah, do you remember that? If we bind IRP to these to the 3' end, we slow down the degradation of the mRNA, and therefore we increase the translation of the protein. Okay, because the mRNA is not degraded because it's blocked. The degradation is blocked by IRP. The translation of this protein can carry on for much longer than it normally would, which increases the amount of the protein. Okay, this is the case, for example, for transferrin receptor. Because we don't have enough iron, we want to increase the amount of transferrin receptor so that we get more iron into the cell. Okay, I'm sure you're going to spend a little bit of time thinking about it. It actually makes sense. It's just a little bit more complicated than...
So if they're present on the 5' end, it's decreasing and if it's on the other side, it's increasing.
That's right. And it just depends on the specific gene, on the specific mRNA, where they are. Okay. So before, when I drew them in one mRNA, that never happens. Okay. You never get a mRNA that has on both ends. Okay. So some genes like allosynthes 2 or ferritin will have them on the 5' end, and transferrin receptor, for example, will have it on the 3' end.
And on the 5' it stops the translation, and on the 3' it stops...
It stops degradation, and therefore it increases translation because it lasts longer. Okay? Yeah?
Could you also bind on the 5' and if you have too much of it, like... Too much
No, because if there's too much iron, the IRP will not bind to anything.
Oh.
Okay? So it's only, the IRP is only active, it only binds to RNA if there's not enough iron. Okay? If there is enough iron, it's in close conformation, it won't bind to anything.
I've got a more general question. I heard that ascorbate is inducing like the intake of iron from in the stomach.
Yeah.
Does that something to do with ferroportin or DMT?
No, actually, well, it has to do with DMT because DMT only transports iron 2. It has to be reduced. So if you eat ascorbate, it reduces more iron, and therefore you can take more iron in. That's the only mechanism. Okay. Well, I mean, I could speculate about some other mechanisms that have to do with IRP. Because ascorbate can actually activate IRP, but that's not it. Okay. It's mostly that it reduces iron in the stomach. Any other questions? If you have any more questions, or if you want to anything explain more, just write on Viuka a question, and I'll try to explain them. Okay. All right.